Hey everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wonder why mapping the brain's connections feels like a wild west of different tools and methods? Well, this new research paper dives deep into that very question. So, what is tractography? Think of it like creating a super detailed map of the brain's highway system. These highways are made of white matter bundles of nerve fibers that connect the different brain areas. Being able to map these connections helps us understand how our brains work, what goes wrong in diseases, and how we learn and think. But here is the big problem the researchers highlight. Everyone's doing it a little differently. And this lack of standardization makes it hard to compare results between studies or even between different labs. It's like everyone using their own map legend and drawing roads in different colors. This paper is all about figuring out what needs to be the same or standardized so we can trust these brain maps more. The authors break down this challenge into several key areas. First up, data acquisition. This is about how the initial brain scans are collected. Even though there are standards like DICOM and BIDS for storing the data, the actual scanning process itself can vary a lot. Different machines, different settings, and even different ways of sampling the brain signal can all lead to slightly different data. Imagine taking photos of the same landscape, but with different cameras and different lighting, the raw pictures will look different. This variability at the start can really mess with the tractography results later on. So, while there are ways to try and make the data more similar, the complex steps that follow can still introduce their own problems. They explain that while we have ways to check the raw scans, it is also super important to check the tractography results themselves. Right now, this often involves experts visually inspecting the brain maps, which can be subjective and inconsistent. A lot depends on what the expert expects to see in a particular tract. They mentioned that numerical checks are better, giving objective scores, but we still need more studies to show exactly how well these checks work and how sensitive they are for finding real issues. This section about spatial coordinates is pretty mind-bending. It talks about how brain images are basically collections of tiny boxes called voxels. When we represent these on a computer, there are different ways to describe where each box is in space and how it's oriented. Think of it like having a set of building blocks, but each block's position and direction can be described in slightly different ways. If these descriptions don't match perfectly between different software or data files, the brain maps can end up slightly shifted or rotated. This might seem like a small detail, but a half voxel shift can lead to incorrect conclusions about brain connections, especially when you are trying to line up tractography data with other types of brain images. This part is fascinating. It discusses how we can study brain connections in animals using methods like track tracing, where you inject a dye. This gives us a kind of gold standard to compare our MRI-based tractography against. But here is the catch: animal brains, even those of primates, aren't just smaller versions of human brains. They have different structures and properties. So, methods that work well for humans might not work directly for monkeys or mice and vice versa. This makes it hard to compare findings across species and even harder to use animal studies to validate human brain mapping techniques perfectly. As brain imaging technology gets better, the amount of data we collect explodes. These brain maps can be huge, taking up lots of storage space and requiring serious computing power to analyze. Traditional methods that work fine on smaller datasets can grind to a halt when dealing with these massive files. The paper talks about solutions like data compression and more efficient file formats. Then to rise the rise of AI, machine learning needs enormous, well-organized datasets to work effectively. If the data is messy, inconsistent in its spatial representation, or poorly documented, AI models can't learn reliably. Plus, there are so many different AI approaches now, making it hard to compare them. It is like having different versions of a recipe and not knowing which one is the best. This is a really important section. Tractography is used in surgery, for example, to help surgeons avoid damaging important brain pathways during tumor removal. But even here, its use is limited. Clinical settings have very strict requirements for speed, reliability, and validation. The variability in research tractography makes it hard to translate these advanced methods into routine clinical practice. Surgeons need to trust these maps implicitly, and right now, the lack of standardization makes that trust harder to build. Finally, the paper touches on how the brain changes as we grow from babies to adults and then as we age. These changes affect the tissue properties and the way brain connections are structured. Standard methods, often developed for adult brains, might not work as well for very young or very old brains. Creating brain maps that are accurate and comparable across all ages is a big standardization challenge. So what is the takeaway? The authors offer a list of recommended solutions. They emphasize standard operating procedures for data collection, using vendor-agnostic tools, and crucially advancing the BID standard specifically for tractography. This is like creating a universal language and blueprint for brain maps. They also call for standardized quality control to catch errors automatically, making analysis more reliable. They suggest making analysis tools more flexible, so they can work with different types of data and across species, promoting fair software principles, findable, accessible, interoperable, and reusable. Of course, dealing with those massive datasets is key, so they recommend building scalable software and platforms. They also highlight the need for shared, well-labeled datasets to train AI models. And importantly, they recommend developing new future-proof file formats that can handle all this data consistently. They stress the need for common benchmarks and metrics for evaluating tractography methods, sort of like Olympic scoring for brain maps. And importantly, they call for building better bridges between research and clinical applications, making sure these powerful tools can actually be used by doctors to help patients. In my opinion, this paper does a fantastic job of laying out a critical problem for neuroscience, the lack of standardization in brain mapping. The challenges are complex, spanning everything from how we acquire data to how we analyze it, and even how we represent spatial information, but the solutions proposed are concrete and actionable. If the neuroscience community embraces these recommendations, especially around data formats like BIDs and robust quality control, we'll be much closer to a future where brain connectivity studies are truly reliable and reproducible. The drive towards more complex analyses and larger datasets, especially with AI, makes this standardization not just desirable, but absolutely essential. The limitations, as the paper implicitly points out, are really around the effort and coordination required to implement these changes across a wide scientific community, but the payoff, more trustworthy brain maps that can accelerate our understanding of both health and disease, is enormous. What do you guys think? Are there other areas in neuroscience where standardization is a big issue? Let me know in the comments below, and if you found this breakdown helpful, please give it a thumbs up, subscribe to Rubin's Mind Lab for more science explained simply, and hit that notification bell so you don't miss our next deep dive. See you next time.